Now, okay, that is a vaccination things. Now I'm going to how the vaccines are development. I'll have only 2 or 3 slides. Okay. So the vaccine development, actually, we're divided into 2 categories. One is empirical, another is a newer category. Empirical, what knowledge we get, what we have that previously done. So based on the things, we are in the development of vaccine. One is a variolation-based vaccine, and another thing is a killed organism and a live attenuated vaccine, and a polysaccharide vaccine, cell culture vaccine. So variolation-based vaccine is a smallpox vaccine, killed organism or polio and other things, and the LAVs as OPV, and a polysaccharide vaccine, and a cell culture-based vaccine is especially for the viruses, say the Japanese encephalitis or rabies, or it is in measles. That kind of is the empirical-based technique. And the most modern type of vaccine is a glycoconjugate vaccine. That is a pneumococcal pneumonia vaccine, a recombinant DNA vaccine by the Hepatitis B, and a reverse vaccinology. That means it's using with a computer application that is called as a bioinformatics. The bioinformatics based knowledge is for the sequencing of the entire pathogen. That sequencing, we are going to find out which one is having the more epitopic predictions, which is having the more binding with that MHC. That also we can do it in our in silico modeling. That is called as the reverse vaccinology. And the next concept is a structural vaccinology. That means a structural vaccinology means that vaccines based on the structure, or protein structure, or is a peptide structure, or it is in a carbohydrate structure. How the structure is going to involved during the antibody synthesis, how is the mutation going to happen? So everything we can get from the structural vaccinology. Next is various kind of adjuvant, that M59 adjuvant, and also is Murale adjuvant and liposome-based adjuvant. So a lot of adjuvants there for enhancement purpose. And also it is a recently we are in the field of nanotechnology. So the nanoparticle-based vaccine also, we are in the field of modern vaccine techniques. One is like a conjugate, recombinant DNA, and the reverse vaccinology, structural vaccinology, adjuvant, liposome, and the nanoparticles. Okay. It is a combine about various spaces there. 6 months back, people saw a lot of news gathered about now vaccine is in a phase 1 trial or phase 2 trial is a PCT trial, like that one. What is meant by clinical trials? Animals, everything. You can get information now here. So PCT means we can establish the vaccine candidate as a desired effects in vitro, in the laboratory condition. So here we are effort to produce a consistently means it can able to produce in as consistent. Same kind of results was happened to the continuous run, like the 1 run, 3 run, 4 run, up to continuous run. So once we got a very good result in vitro, we are moving to the in vivo. That is in a animal studies. In vivo. So doing the animal studies, we'll get some desired immune responses. It is able to protect animal from the diseases. Yes, we are going to inject to the animals for the antibody development. After the antibody development, we are going to challenge the particular pathogenic viruses or bacteria. So it leads to the protection. That will be in animal studies in vivo. Once it's completed, we are going to the human volunteers as a phase 1, as a less than 100 volunteers. Here only we are doing for escalating the doses. Escalations means we are going to the, that time only we are going to concentrate on the 0.5 ml as a dose or 0.1 ml dose as 1 ml as a dose based on the results what we are obtained from the blood samples in phase 1. So once the successful completion of the this one, phase 1, we will move into the phase 2. It's like 100% volunteers, sometimes 1,000 people, so we are going to use that. And intended based on the age and the diseases. Okay, we are going to categorically, as a based on age, based on the gender, based on the disease, and also during this phase, we are checking out the additional safety and efficacy testing. Efficacy means how much our vaccine going for effectively or potentially or immunogenicity. So everything studied in here at this phase 2 level. Once it's cleared, we are moving to the phase 2, phase 3 with a higher, that is a higher amount of person. Here they expanded our safety database. So here expanded safety database means, so each and every person, those who are in the participated in the vaccine trials, the entire detail we are kept for the safety data database for the approval from the organization. And also they establish the protective efficiency, the clinical consistency, moving for the market releases. Then next to that, phase 4. During the phase 4, once a vaccine successfully going for the phase 3, going to the post-marketing surveillance and detect the adverse effects and assess the long-term efficiency. So these are the category for the vaccine efficiency at various phases in the vaccine trials. Okay, that's the vaccine trials. Here, various techniques are involved in vaccine development, various vaccine development. One is a recombinant DNA vaccine subunit. How the subunit vaccines was developed. First, we are going for the isolated the viruses, and after the isolation, we are going to extract the DNA. That means the DNA means how it's going to give a DNA is a blueprint marker. Then after the DNA isolation, the sum of the gene only responsible of the particular surface gene. Either it's a surface protein or other thing. So particular gene we are going to analyze through. We are using with that the reverse vaccination technique here. Next, moving to the non-pathogenic vaccinoviruses. That viruses we are going for the vaccinovirus extraction, RE digestion, and final ligation to this vaccinovirus type. Okay. So this thing, we are going to transform it to suitable host, then upstream process, downstream fill and flesh. So here, this kind of is a vaccine concept. It was currently available like mRNA-based vaccine or adenovirus extraction vaccine, Sputnik vaccine also in same kind of category. So this is isolation of virus from the COVID-19, from the extraction of DNA from the entire nucleic acid. Their decide gene, that is the mRNA responsive surface gene was transmitted. Immediately they are going for selected a vaccinovirus strain for the non-viral replicating vector. So after replicating, they're going for transform with a suitable host, then upstream, downstream, fill finish vaccine. So these kind of techniques, you can see that those vaccine available in the market against COVID-19. So here you can get some idea about the pictorial representation of the recombinant. So DNA extractor, herpes simplex viruses. So from the DNA, we are going for isolated, then going for gene isolated, that gene specific for the surface protein. Simultaneously, the harmless vaccinoviruses. That's only carrier molecule with a transfection, infection through transfer of our gene. So harmless then vaccine DNA is going for digested that the fragment containing surface gene, and in the host source, it is not causing the diseases, but only inside what we have the surface protein is going to inject to the person and release to the antibody. The recombinant DNA vaccine is a subunit based to that. And another vaccine is a recombinant hepatitis B vaccine. So the hepatitis B vaccine, the hepatitis B viruses is a DNA or RNA genetic material. The antigen producing genes are cut here. After the cutting, we are moving to the selection of the desired vector, like the bacterial DNA, as is the plasmid DNA. So that plasmid DNA we are cut with the restriction suitable endonucleases, RE digestion. So this gene going for recombinant DNA, going for transformation into the recombinant vector. After the recombinant yeast cell, we are going to recombination for higher cell culture. So inside the fermentation of the yeast is going to secrete something. That is the hepatitis B protein, multiply and produce H2 antigen in a fermentation. From the fermentation, we harvested the culture containing hepatitis B protein. That protein we are moving for the further extraction and purification. That is called as a hepatitis B vaccine. That is a commercially available vaccine now. Okay, so that is a vaccine. Now we are moving to the production aspects. The production aspect means rabies. I'm going to the what I told is what is vaccine, what is the vaccination, what is the type of vaccine, how is it going for development, why we need a vaccine. Now I'm going to the how the vaccine was produced in our past institute, and also how the technique was used currently. What we have the co-vaccine basis. So that kind of things you'll get information here. So before that, I go for giving some clarity about rabies diseases. Rabies, it is a zoonotic diseases. That means even COVID-19 also zoonotic. MERS, SARS, and Crimean Congo, and cancel forest disease, Sintara, and a lot of viruses, Nipah. These are the zoonotic viruses. Zoonotic means there's a disease that is transmitted from generally that rabies disease was rabies viruses. It's under the group of Lyssavirus genus within the family of Rhabdoviridae. The virus is transmitted in the saliva of rabid animal. So what rabid animals means, those animal got the rabies viruses, that animal is called as a rabid animal. And generally, that's DNA. Sorry, generally that saliva, it is entered not through the skin. It's going for the infiltration by the virus-laden saliva from the rabbit animal. How it's moving made by the dog, or at least something scratches is going to correctly restrict exposure to the mucosal surfaces. That means when you're going for inhaling the saliva, if the saliva having the rabies viruses, going for the bite. Once virus was entered to our body, it is going to the 100% fatality. 100% fatality means if a person having rabies virus, there is no further treatment. If a billionaire or any time, there is no such a medications available. So there's 100% fatality, but it preventable. Even though we can control, we can preventable. But how? By the vaccination. So mostly, it is a 95% was happened by the dog bite. Sometimes it's less than 4% of monkeys, donkeys, horses, and cows, buffaloes, goats, sheep, and pigs. Occasionally, in less than 1% is wild animals like mongoose, foxes, jackals, camels, and elephants. So these are the causative agents. Rabies is zoonotic, and rabies is caused by rabies viruses, and is transmitted saliva through the infiltration by virus laden, through the scratcher or direct exposure. It is 100% fatality, and also it is the most common, as 95% is in dog. Some of the things it will be in like donkeys and other horses. This is about rabies, epidemiology, and affected rates. Then how can we identify the this thing? There is a rabies in animal. Like rabies in animals are furious and paralytic, rabies, apparent rabies, and a rabbit recon. You can see some what do you call it? It's a different kind of dogs that are having that various types of salivas is coming out. It's a first dog. You can see that it is called a furious stage. It's going to bite others, a tendon to bite others. And another dog, you can watch this. Some saliva is coming now. No, this saliva. It is in the saliva. It is in. It is called as a white color salivation coming there. And the next is a dog. It's having and also cunning like dog. And another dog is going to bite you. Saliva drooling. Saliva come out as a more number. So next is that raccoon. So these are the main culprit to the causing the dog bite. Okay. How the rabies virus is going to transmit it here? You can see that some dog, pet dog. It look like it's a pet dog, and also it having the some rabies viruses. Okay, here some of the student they might be have some pet animals, and also if a person having that kind of that kind of accident maybe happened earlierly. So once they went to the hospital, they asked about please watch the dog. Why doctors are advised to watch the dog for minimum 10 days after the bitten to the person, because that dog going to die. How the dog going to die? By the transfer of rabies virus to the dog brain. Okay, whether any clue we can identify our pet dog having rabies or not. That means yes, based on my expertise in that rabies identification. Whether you can see that this dog, the white color saliva, if dog having based on the color of saliva and also density of the saliva, if the dog having the white color saliva, it is in a highly watery, not in a viscosity. That means, and also some viscosity is there, means that that saliva, it is not containing any kind of rabies virus particle. Here I told the rabies virus particle. That means it is not a rabies intact material, and also some virus particle will be there. And next, the same saliva is color going to the pale yellow. That means 50% of the viruses particle present in the saliva. And the next is a pale brown. If that dog saliva having the pale brownish color, it means that person having the dog having the 100% of the virus particle present in the saliva. What the saliva containing? Rabies viruses. That rabies viruses, it been bullet-shaped virus. Already I told in my first slide, it's a bullet-shaped rabies viruses. Yeah, it is there. The bullet-shaped viruses, it is called as a different kind of structure protein. One is a bullet. It is a outer core membrane is called as a glycoprotein and lipid envelope, matrix protein and ribonuclear protein. So all kind of proteins are present as a structural protein, and other non-structural protein also there for a particular function. And this virus is how it's going to transmit a person. You can get some information here. Here, a dog is going to bite a person. So the virus was inoculated Earlier I told through the filtration. So once inoculated, there is an opening was happened on the skin. So through the saliva is going to transmit it there. After entering, the virus replication was happen in the muscle. Once the muscle means it's directly not on the muscle, it's going to the contact with that acetylcholine receptor or dopamine. That's the receptor. That is a neuromuscular junction. But based on the things, so it's moving there. It's moving to there, and the virus travels within axons in a peripheral nervous via the retrograde fast as well axonal transport system. Once attached to the dorsal root ganglia as with the spinal cord, the replication motor neurons on the spinal cord and the local dorsal roof ganglia and the rapid ascent to the brain. So rapid ascent to the brain. Once it crossed to the blood-brain barrier, so immediately the infection of neurons with the neural disinfections, so multiplication is much more when compared with other part of our body. So immediately is going for dysfunction of the brain system. So everything stopped because of the rabies virus multiplication. Yes, the virus cannot infiltrate into the skin. Once the virus reaches the brain, it further replicates, resulting presentation of the clinical signs from the patient. What are the signs? You can see this kind of thing. Okay, one is hydrophobia, and also photophobia, and aerophobia, spasm, and you could not able to was, and finally going to die. Why it was happen? How it was happen? Yes, so brain. What is the main function of brain? Brain is a control our entire system for walking and moving, any kind of activity on seeing and digestion, everything. Here, brain is not properly functioning. Whether the person he can able to walk, still no, because there is no signal, there is no proper signal for the brain, so he could not able to walk. So he'll be only in lay down positions, or it is in moving in the forelimb, like it. And the next is high cone cell rod cells. You know the better. Okay, that cone and rod cells, it gets some signal from the brain. If it is not a proper signal, but signal is coming, if it is not a properly not going to able to see anything, leads to the photophobia. Like hair also is not good to be hearing anything. So hearophobia. And the next is the esophagus. The esophagus is the main function towards that what the materials we are eating. It is a solid or liquid, everything transport to the intestine. So that esophagus is made up of, that membrane is made up of striated and non-striated muscles. Going for that relaxation and contraction there. That relaxation contraction also get a signal from the brain. If it is not get a signal from that, what will happen? So immediately those contracted things is not relaxed. That person he could not able to eat anything and also not swallow, could not able to drink anything, leads to the hydrophobia. And the spasm leads to happen. So and next to that, nearby the esophagus, we have the another organ, that is the sharing